Hey gang, and welcome to another exciting episode of Biology Online. And today I'm doing something a little different. I'm going to walk us through how to do a Punnett Square and kind of the method that I use that helps me do better on it when I was learning to do it. And I'll also probably go ahead and walk you through a monohybrid cross and also a dihybrid cross. So to get us started, here's our Punnett Square. And I've already given us two different genotypes, one for each parent. I'm kind of modeling this example off of Mendel's experiment, so I'll probably talk about it a little bit as I go on. But here we have a homozygous dominant and a homozygous recessive. They're homozygous because they're both the same and they're both dominant, so therefore it's homozygous dominant. These are both the same, they're recessive, so they're homozygous recessive. And what we're going to do is we're going to breed these two, or crossbreed these two individuals together in this Punnett Square. But let's talk about how I actually go about doing this. So whenever we have those two letters, remember that these letters represent chromosomes, right? More specifically, a trait on that chromosome, but we're still talking about a chromosome. So there's a reason why we have two letters. There's one chromosome from mom, and there's also the homologous one that we got from dad. So there'll always be, in most cases, there'll always be two versions or two alleles of each trait, because we have a version from mom, we have a version from dad. And so we always need to be able to represent them in pairs of twos. And the letters simply represent the traits. The different letters, different traits. So if it's the same letter, it's different versions of a trait. So in this case, big R, I think in Mendel's experiment, was purple, and little r in his experiment was white. You'd probably think, well, it's two different colors, why aren't there two different letters? Well, it's not the color, it's the trait. There's two different versions of the color trait. We'd say there's two different alleles. So when we start talking about how my process for doing this, I actually kind of have to go back and think about these as chromosomes. And whenever we reproduce, whenever organisms reproduce, especially ones that reproduce sexually, they have to go through meiosis before they can do this. All right, so I'm going to rewrite these letters here. So here's a big R, and here is another big R, right? We need to figure out from this parent's genotype, right? Here's the one they have for mom, here's the one they have for dad. They just happen to both be the same allele, the same version of the trait, in this case purple. We're going to take this individual through meiosis in order to determine what type of gametes, what genetic combination of gametes this individual can produce. So the way I do this is I underline the trait, right? Then I draw a line in between them. Underlining each trait helps me just keep them separate, because later on when we do dihybrid, there'll be more than just one trait. So that we may have big R, and then but later on we have big R, and then like big A. I'd underline the R's and I underline the A's to let me know that those are separate traits, right? Then I draw a line between them, right? And that line between them, for me, just represents meiosis. Right? So this is meiosis. And in meiosis, the whole point of meiosis is to reduce the number of chromosome down to haploid. Because right now it's diploid. Our diploid number would be 2, because there's 2 chromosomes that we're talking about. So if we go from the diploid number down to the haploid number, 2 should be divided down into 1. That tells me that each sperm or each resulting egg or spore or pollen or whatever it is you're making should only have one of these chromosomes in it, not both. So what could this parent give? Well, they can give a big R from here. What is that with me today, and drawing R is funny. Big R from there, right? Or it can give this big R, right? Me learning how to draw with a mouse. Big R or big R. This will become a sperm or an egg. This will become a separate sperm or an egg, right? Let's look at the other parent, right? We had little r. Underline the trait, draw a line in between that represents meiosis. What can this parent give? Well, from this side it can give a little r, and from this side it can give a little r. So that would become a sperm or an egg, and that would become a separate sperm or egg. I'm essentially going to, now that I know what sperm and egg combinations genetically can be given by these adults, I'm going to write this on my my Punnett Square. It doesn't really matter which one is which adult. I'll just do the big R's for female, or give her the homozygous dominant genes. So big R, and I'll give the male little r, right? And just, this is the female symbol. This is the alchemical symbol for Venus. The cross at the bottom represents the womb. This is the alchemical symbol for Mars. The arrow off to the side represents, it's phallic, so it represents a penis. So, that's the Austin Powers symbols, as some of my students tell me. So that's the female, and this is the male. So I'm just putting the different combinations they can make on each side. So in this case, monohybrid cross. This would be one gene she can give, or that. And she can't give both, right? She can't get 2 R's on this side, 2 R's on that side, because we go through meiosis. We split the genes apart, right? We cut the number in half, go from diploid down to haploid. So now to fill in the Punnett Square, it's easy. We just take what's on this side and we bring it across, right? And we take what's up here and we're just going to drop it down. There's a simple rule for writing these things down to help keep them clear in our head. I'm always going to write the dominant letter first. So if there is one, I'm going to write it first. I'll put my R's down. You can do it per block. I'm just going to go ahead and write them all down real quick here. Then I'm going to bring whatever's on this side across, and then in this case it is the little r. Bring him across, and then this guy can also give a little r, so bring him across, little r and little r. So always write the dominant letter first. A lot of people want to come in and they want to write little r and then put the big R next. Now technically this isn't incorrect, but if I'm in a hurry and I'm trying to counter this stuff, it's easy with a monohybrid. We only have 4 squares to fill in, but what about a trihybrid cross? There'd be 64 squares to fill in at most. That's a lot to go through and count, and there's a lot of letters. When I look at this, if I'm in a hurry, the first letter I see is the little r. I may think this is the recessive trait, and that's what's going to show up. But there's a big R sitting right next to it that I may have skipped simply because I saw the little r first. I always encourage you to write the dominant letter first for that trait. So, and we've done that. So let's look and see what we got. So this individual in Mendel's experiment was purple. Almost had been purple, and this one was white. All of his generations had been white, so he called them pure for purple, pure for white, and he crossed them. But the result that he got, no matter how many times he bred these together, he always got individuals that showed the dominant trait. They always ended up looking purple. 0% of the flowers were white, and that baffled him. He knew that hey, this one only had white to give, so I know the white is in the kids someplace, it's just not showing up. And so he did something kind of brilliant at the time. He first of all, he named these generations, right? He called the original parents, he called the P1 generation. The resulting offspring he called the F1 generation. P1 stands for first parental, the F1s stand for first family. He crossed 2 of the F1s. He crossed this guy and that girl. All right, and he got the next resulting group of offspring he called the F2s, the second family. But when he crossed a big R little r with a big R little r, he ended up being able to find those recessive alleles or the white alleles, and he's like, hey, I knew they were locked up in there somewhere when I crossed 2 of those offspring to get the F2s. Genetically, he came out 25% of the time he was getting that white allele showing back up. It was hidden in the F1s, but it reappeared in the F2s, and that led him to the discovery, hey, some versions of a trait, some alleles are recessive and some are dominant. The dominant ones, if they're there, will only show up and be expressed. The recessive ones will always can only be expressed when there is no dominant gene present. And this was a big breakthrough. For the longest time, people knew that, hey, here's a fat pig and here's a fat pig. Across them together, I'm going to get fat pigs, right? Except for every once in a while, they got a runt. There's different conditions that can lead to a runt, but there's also recessive traits that can lead to a runt that you don't see in the adults, but may show up in the offspring. And he really helped the understanding of that. Now, I will say it's just kind of a default that there's no gene in the human body. The more that we learn, the more that we discover there's not a gene in the human body that actually works off a simple dominant recessive. There's a multiple interplay between multiple genes, multiple chromosomes that lead to the expression of a certain gene. So it's not as cut and dry as, hey, I'm dominant, I'll show up. There's other things that are going to affect the expression of genes. And the more we study it, the more that we learn, and that's really cool. But it's still based off of this understanding of simple dominant recessive. So yeah, so that's pretty much how we do the basics of doing a Punnett Square. Again, it helps me to underline the trait and to divide them to remind me that you'll never end up with two of the same letter, two R's, either capital or lowercase. You'll never end up with two R's in any of the resulting sperm or egg because we have to go through meiosis. Just to kind of help you, just a fun thing to help you remember this. When I was a few years ago, when I was teaching, I was in Arizona for a little bit of time teaching, and one of the students there, when I was showing her the class this, it was mostly nursing students. So it was very heavily populated with females, and one of the older women in the class says, "This kind of reminds me of a bra." And I kind of paused for a second, and I was like, okay, I'll bite. You know, what do you mean by that? And she says, it lifts and separates. And I was like, okay. So always remember to give your genes a bra. So underline the trait and separate them to let you know that, hey, this is one trait and it goes through meiosis. So neither side should end up together in the resulting sperm or egg. I hope that helps you understand it a little bit better.